I see and notice that we're now streaming as well. Very
We
good.
are. Okay, I think we'll start to begin so that we remain on schedule. So, hello, and welcome to this online synthetic biology forum organised by the University of St Andrews iGEM team of 2020. I hope that you've all had a good week. If it's been a particular busy one with work or other commitments, then this webinar provides a more relaxed end to the weekdays in preparation for the weekend. My name is Bradley and I'll be your host this afternoon. We also have Yashwan, who will be your co-host. If you would like to wave and greet, Yashwan.
Yeah, hi.
We have Caius, who will be dealing with any questions you have throughout this event.
Hello.
We have Lawrence, Sue and Polly, all respectively as well, who would all like to wave and greet.
Hi everyone.
They'll all be assisted in their own way to make sure the event is engaging and enjoyable for all of you. The purpose of this forum is to introduce you to the field of synthetic biology, to those members of the audience who may not be so aware of it, and more specifically looking at the topic of GMOs, that is, genetically modified organisms. The term itself gains quite a lot of media attention, and to many people it has connotations of killer sweet corns or apples on drugs. But what exactly are GMOs and how can they be used in modern day society? We hope that this online forum provides a free platform where the public can be educated on the topic from credible sources, and questions like this can be reliably answered. So without further ado, I'd like to introduce our speakers as our panel of experts. We have with us today our very own Chris Hooley, Senior Lecturer of Condensed Matter Physics at the University of St Andrews. We also have Professor Les Furbank, a Professor in Sustainable Agriculture at the University of Leeds.
Hi there.
We have Dr. Louise Horsfall, Chair of Sustainable Biotechnology at the University of Edinburgh.
Hello.
Slavena Todorova, a chemistry teacher at UCHA and a laboratory specialist at iGEM.
Hello.
And finally, Dr. Richard Harbottle, Head of the DNA Vector Research Group at the GKFZ Cancer Research Institute in Germany.
Good afternoon.
I'd like to thank all of them for agreeing to participate in the forum today, and what a privilege it is to have them on here. So before we really get started, I'll provide a brief explanation of how the event is going to proceed today. To begin with, we're going to have our very own synthetic biology expert of the team, Caius, give a short background on the information of the topic. And then we'll have more formal introductions from the panelists themselves, who will discuss in more detail about who they are, what they do, and if time permits, address any questions you may have for them in particular. This should bring us to the turn of the hour, where we'll have a short break, and then the panel discussion will begin. We really want to have as much engagement during this event as possible, and so we encourage everyone to interact with each other through the chat channel and ask any questions you have via the Q&A. As I've mentioned, Kais will be handling these for yourself, and where it is possible, you'll type answers to your questions, but we're specifically directed to the panel experts themselves, or it's more appropriate, we'll try to answer this live, and we'll give you the opportunity to speak to the panelists directly if that's preferred. However, I'd just like to mention at this stage that the event will be recorded, so if you're uncomfortable with being in the audio of this recording, then just please ask us and we'll try and speak for you. Of course, we may not be able to answer all of your questions during the event itself, but if any questions are dismissed or left open, then we'll be sure to follow these up after the forum by contacting the questioner via email and posting the answers on our social media platforms. If you feel someone else's questions particularly good, or you're wondering the same thing, then you also have the ability to upvote this question during the panel so that it appears at the top of the list. Indeed, there will actually be prizes at the end of the forum for the best questions, so get questioning and get voting. Furthermore, feel free to comment on other people's questions if you think you can be of help. This will still keep the questions open for us to address. We do realise that not everyone may be familiar with the Zoom platform, but you should all have a toolbar at the bottom of your screen with the ability to submit questions in the Q&A, chat to others, and also to virtually raise your hand. Please use this if you need any help in any way during the event and one of our team members will try and assist you. I believe Polly will post a link to this with a more detailed guide in the chat for those that find it useful. Okay, so I think that's me for now, and I'll pass you over to Kais, who will give you a brief introduction on the topic of discussion today. Will GMOs shape the future of humanity?
Well, thanks, and welcome for coming. It's certainly a strange event to have this in the midst of COVID. So, I mean, essentially, synthetic biology is quite a new discipline, and it really has its modest origin in around 2000, with the publication of the repressilator and toggle switches, which were two rather revolutionary gene circuits, so the connection of different genes which together serve a certain function, like the different parts on a circuit board. And they, from there, really lit up the landscape of genetic engineering. It paved the path to turning biology into an engineering science, really in order to decode the raw computing power of a single cell. And at the same time, actually establish an open source community, uncluttered by paywalls, expensive lab costs, and patented techniques. So at last, there really is no escape from maths and biology. In a sense, its success is tied very closely to iGEM, the International Genetically Engineered Machine Competition. And really, as one of its main proponents of synthetic biology, and it started in 2003, so quite shortly, just after the real start of synthetic biology, generations on generations of senior school and university students compete by designing and implementing, both in the lab and on their desktops, novel solutions to different environmental, medical, and pest-related problems. So part of that is this open source drive called the Registry of Parts, which is an immensely large database of regulatory and genetic DNA. So both the elements of promoters, ribosome binding sites, and different DNA regions that code for different genes from hundreds of thousands of different organisms, which, like a clothes catalog, you can really just cherry pick them to be sent directly to your lab to be copied and pasted into the cell. And in a sense, that's the revolution of synthetic biology, to reach a stage whereby you can simply transcribe across any discipline in the sciences, take a gene from one organism and convert it into another, as you would copy and paste text from one script to another. So for example, examples range from E. chromai, which was a Cambridge project which tried to change the color of feces, your own feces, depending on the bacterial and digestive tract. So it would turn red, for example, if you had a particularly bad E. coli intestinal infection, and yellow if you had a lot of lactobacillus in your tract, for example, to splash, which is a rather novel antimicrobial bandage to combat burn wounds. So really the range of projects is quite limitless. So again, despite the burgeoning advances in biotechnology and the growth of genetic engineering community, controversy still heavily surrounds its use, particularly in genetically modified organisms, GMOs. And for some, they might associate with BT corn, golden rice, and even the flavor saver tomatoes. But for others, there are the unpredictable consequences that they feel come with genetic engineering, such as off-site mutations or unpredictable effects when they're introduced into the environment. So although genetic engineering, synthetic biology, and GMOs aren't synonymous, they do still converge on the same technique, gene editing, albeit with different goals. So hopefully this panel will take us through the challenges and controversy, the excitement and excellence that come with of course, synthetic biology, GMOs, and also the possible ethical and biosafety concerns that surround their uses. So in the end, why do scientists want to use GMOs, and what are the arguments against it? And importantly, what can GMOs be used for? Hopefully this brilliant panel with, again, fields as far-ranging as condensed matter physics, all the way to agriculture, shall be able to provide insights on the different approaches and the modeling steps that can be taken in order to see their impacts in real life. So thank you very much. And I think without further ado, I think we'll start our panelist introductions. So maybe if we begin with Leslie Furbank, and you'll just see now that you have a poll that you can answer.
I'm just moving the poll out of my screen so I can see things. Thank you very much for that. No pressure. I confess I went to university a long time ago with the intention of studying biochemistry, so I would understand a lot of this sort of stuff, and promptly fell asleep. Instead, I became an ecologist because I was sold by the idea. Basically, the first lecture I went to in ecology was an hour-long slideshow of all the places that you could go to as an ecologist. I thought, I can do that. Now, my background and my interests until the late 1990s were the ecology of agriculture. In other words, how changing different farming practices would impact the environment, and in particular, impact on wild species and biodiversity. And when we got to 1999 in the UK, we'd had the first GM crops commercialized in the United States, and they're being developed to be either resistant to broad spectrum herbicides. So in other words, you could kill the weeds more effectively, or else they would have basically inbuilt pesticides. They were never going to be relevant to the UK because we didn't have the pests. But there were moves to introduce the herbicide tolerant crops. But there was a lot of controversy at the time. And the main controversy was that although they'd been tested as being safe in terms of are they safe to eat, they hadn't been tested in terms of their knock-on effect on the environment. And there was a lot of concerns at that point about the impacts of farming on particularly farmland birds. So I was fortunate enough to end up leading a project to look at this. And what we found after, well, at the time, it was the biggest experiment ever done, certainly in ecology. And what we found was that, yes, there were differences between GM and non-GM crops of the same type. But they weren't due to how the crops had been bred. They were simply due to the herbicides that were used with the different varieties. Secondly, we also found that the differences between crop types were bigger than the differences between GM and non-GM. In other words, what we found was that the range of variation between GM and non-GM was within the standard range of variation of agricultural practice anyway. What also turned out was that of the crops that we looked at, two of the crops, which is oilseed rape and beet, the GM actually had less biodiversity in there. And we're talking about less insects, fewer plants. Whereas the maize, it was the other way around. And this again was due specifically to the herbicides that were used on the two different treatments and nothing to do with how the plants had been grown. So that got me very much into the whole area of the environmental risk assessment of GM crops. And I'm continuing that, not in terms of research, but I'm on the European Food Safety Authority, which undertakes the risk assessments on behalf of the EU. And where we're particularly looking at the moment is that we do have an opinion. In other words, this is a statement that's going to go out on our journal on synthetic biology and whether the current regulations are fit for purpose. And the area that I'm looking at in particular is whether gene drives need to be assessed more carefully. And the gene drive is where instead of releasing a crop or something like that in a fairly controlled environment, the idea of giving an organism a gene drive is that the drive will drive a trait against natural selection and into wild populations. And that's particularly being developed with the idea of either reducing mosquito disease vector populations, or more interestingly, changing them so they no longer carry the diseases. So over to somebody else.
Thank you very much. Firstly, do we have any questions from the floor? Okay, not that I can see. So I think that was very fascinating to hear about your work. So maybe an interesting question would be, so as a member of the FSA, what are the various principles surrounding regulation of GMOs, and what factors are taken into account directly?
Well, first of all, one of the key things is it's a case-by-case approach. So in other words, every case has to be addressed individually. And it's not just a case of whether it's a particular kind of GMO that's been looked at. Now that is different from other regions. Sometimes, so Canada looked more at the kind of GMO that's been looked at. Secondly, we're only concerned here with organisms that are released into the field. So we have nothing to do with lab-based or enclosed organisms. So in other words, we don't address any of the issues in GMOs used for medical purposes, for instance. But one of the trickier ones that people find it hard to get their head around is the idea is that we must, in combination with whoever it is that's developing the GMO, is look at the risks, the plausible pathways to harm. In other words, if we have this particular crop, for example, what kind of harm might it make? It might be allergic to people. So in other words, we need to find evidence that that's the case or not the case. There may be various environmental impacts. What are they, and what evidence do we need to actually weigh that up? So it's not about generic, unfocused concerns about harm, but what are the real worries, and actually is there evidence as to whether those worries are well-founded or not? And if the answer is well-founded, then it's quite simply the GMO is not allowed to be released. Also, EFSA, and this is the same with the UK authority ACRE, we do not decide, we provide the science evidence for politicians to make the decisions. So that's very important. We provide the evidence input, but we do not make the decisions.
Okay, thank you. So this is a question that was asked by an anonymous attendee from the floor. And it is, do you see gene drives being used in pest or vector population control in the next 10 years?
There are several major groups around the world that do see this, and there's a lot of research going on at the moment. The particular focus is looking at gene drive mosquitoes for the control of malaria, especially in Africa. I wouldn't want to comment as to whether that is actually going to happen, but certainly there is a lot of effort and resource going into that one.
Oh, and I think we still have time for one final question, if you don't mind. And that's
I don't know about Florida and Texas, but I've not heard of any efforts to use the United States for gene drive releases. But that doesn't mean to say it won't happen.
Okay. Well, thank you again for your introduction.
Yeah, thanks very much, Les. I think we're right on time, and we just have time for our next introduction from Louise. So without further ado, I'll hand over to yourself, Louise.
I'll unmute myself before trying to talk. Okay.
That would help.
Yeah, I almost did it then. Okay, so yes, I'm recently Professor Louise Horsfall, and I work at the University of Edinburgh in a large interdisciplinary centre that we have there called the Centre for Synthetic and Systems Biology, Synthesis. So my interest now for a number of years has been about applying synthetic biology tools and approaches in the area of biotechnology to make it more sustainable. That's quite a broad umbrella, and I'll give you some examples. So I lead a research group in this area, and I'm also programme director, so I'm responsible at the University of Edinburgh for the postgraduate teaching in synthetic biology and biotechnology. I've been involved in iGEM for a number of years in a number of different ways. I'm currently not responsible for the Edinburgh iGEM team, which I have been in years past, but that doesn't stop me being involved in iGEM. I'm currently part of iGEM's Diversity and Inclusion Committee, which is a great pleasure to be a part of. And this year we're going to be awarding a prize in this area for the first time, which is fantastic. So I am the Royal Society of Edinburgh's Young Academy of Scotland's theme lead for sustainable or sustainability. I'm also an elected co-chair of the European Federation of Biotechnology's Bioengineering and Bioprocessing Division, and I'm an editor of the Microbiology Journal, that's the lead journal of the Microbiology Society. So I've got quite a lot that I'm busy doing, which means that the people that actually do all the work that I'm about to tell you about are my research group. So they're just introduced here. I haven't identified any particular of our research projects with any particular researcher. There's a lot of overlap, and I'm only going to give a few examples of the work that we do in my lab. But as I said, we're very much focused on trying to make biotechnology more sustainable. And there's a number of ways that you can do that with green technologies, clean technologies, all these words that we're currently using. It can be about thinking about green energy, biofuels, but alternatively, what we're trying to see how biotechnology can actually help us to move from the current linear economy which we're in, which we take, we make, and we throw away, and transition into a more circular economy. And can biotechnology help us do this? But also, I think of increasing importance is actually how can we make sure this emerging bioeconomy is circular in itself and not linear? Because we all know the processes that we're developing, you grow a load of bacteria and they do fantastic things for you, and then you pull them down the sink after you've killed them with zircon. So it's trying to make as much circularization as possible. So not thinking about that part, thinking about a circular economy and how biotechnology can contribute to it. One of the things that my group is doing is looking at metal recovery through biological means. Now, I don't know if you're aware, the UK is striving to massively decrease its carbon emissions. It's got a number of timelines to make sure it does this. And those timelines can only be met, I mean, there's argument over this, but they can only be met if we transition to electric vehicles. There's an awful lot of research in this area going into lithium-ion batteries, in particular how they can be used in transport. But that not only means that the demand for the raw materials to make the batteries is exponentially increasing, but also at some point in the future, we are going to end up with an awful lot of used dead batteries. And currently there is no method for effectively recycling them. So we're working as part of a UK-wide consortium to work out how we can recover, remanufacture and recycle the components of lithium-ion batteries, of vehicular lithium-ion batteries. And surprising to some, perhaps, that biology has something to offer here. So biology is fantastic in its selectivity and its sensitivity to metals and its ability to tell the difference between different metals. And there are challenges there are challenges being posed by the current composition of lithium-ion batteries within this recycling process, such as cobalt and nickel are quite similar and quite difficult to separate. but biology can do this, and it very easily can tell the difference. So what we're trying to do is use both biotechnology and methods to be able to screen bacteria for their ability to selectively recover metals, and for example they produce them as nanoparticles here so that we could recover them, but then to actually use synthetic biology to enhance what nature can already do so that it can be turned into an actual industrial process. So that's how I mean we employ synthetic biology for biotechnology purposes. So that's one example in our biological metal recovery that we do. What we're also trying to do is perhaps it's very similar, but it's also upcycling. So taking industrial waste streams with regards to the batteries, that didn't come out very well. The proposed method is actually that those metals will be reused in lithium-ion batteries. Whereas we have an awful lot of industrial waste streams, historical legacy waste streams, and also current waste streams that again are contaminated with metals. Land is contaminated with metals. Various different waste streams have metals dissolved in them, and we're using synthetic biology and bacteria to be able to recover those metals. We're using synthetic biology methods to engineer the bacteria so that we can actually tailor the nanoparticles that they're capable of producing for alternative purposes, for remanufacturing, so that they can be used in materials in the future. Other things that we're doing within this very broad sustainable biotechnology is trying to enhance the biological degradation of lignin. So lignin is a recalcitrant bio macromolecule. It's at the moment it's massively available. It's a waste, and it's would be fantastic if we could actually degrade it to a consistent form that we were then able to make them use the carbon that's stored within it. We're also looking at how we can engineer bacteria so that they can be used better, so that they're more useful within industrial processes. For example, we've isolated some novel industrial anaerobic bacterial strains. We're characterizing them, and we're also trying to develop a toolkit so we could engineer better acid resistance within them, so that they would be more productive because they could survive the conditions that are presented within an anaerobic digester for longer. So I meant to start my timer and I didn't, so I have no idea how long that took. So just to sum up very quickly, lots of things that we do in my group are to help use biotechnology so that it moves us to a more sustainable and circular bioeconomy, and that fits very much within certain parts of the UN Sustainable Development Goals. Thank you.
Well, thanks very much for that, Louise. That was incredibly interesting. I think we do have a couple of questions which we might have time to answer. Have maybe one of them. So one from Sarah Welch. Would you like to ask this yourself, Sarah, or would you like Kai or myself to ask it for you?
Okay, maybe we'll simply say the question for you. It's fine. So the question is, how do you work with anaerobic bacteria?
So there's there's several different ways. Within the lab, when we're actually doing the engineering, we have to use an anaerobic chamber. So we do have a chamber that gives us the anaerobic conditions to be able to grow the bacteria in. within an anaerobic digester, it's and at industrial scale, it's really not very difficult to have anaerobic conditions. What you actually have to do in a fermenter is oxygenate it all the time, because that's the problem, getting the oxygen in. It's just when you're working with the tiny quantities that you use at lab scale when you're doing the engineering, that's when you have to use the anaerobic chamber.
And just one query regarding the process whereby you are screening bacteria for metal resistance and therefore the ability to form metal nanoparticles. Did you do this in an evolutionary process where in each stage you would increase the level of metal toxicity perhaps and screen at each stage?
So that's definitely something that we have tried with a bacterium that we already knew produced the nanoparticles. So that in itself is quite a big step. Just because a bacterium is quite resistant to metals doesn't mean it can produce nanoparticles. So you need to find one that produces nanoparticles anyway. So that's kind of one of the screens. And that's expensive when you actually start using the electron microscope to be able to see the nanoparticles. So what we have done with an organism that's capable of producing nanoparticles, we've done a 2-year experiment of increasing the metal that we expose it to, regularly subculturing this for 2 years, and we found that it did not enhance its ability to produce nanoparticles. That was a lot of time and effort for that experiment.
Thanks very much for that, Louise. We have got another couple of questions, but I'm going to move on to our next introduction speaker. So maybe if yourself, Louise, or Kais, if you could try and answer them via the Q&A chat, that'd be great.
I think these will also be some excellent questions to ask to the greater panel when we finish the introductions.
Of course. Okay. So I think next up is our very own Chris Hooley from St Andrews University.
Yeah, thank you very much. So let me start as Les did at the beginning of my university education. I actually went up to university to read physics and philosophy, a joint honours degree, and it was the philosophy I was most interested in when I began. Over the course of the 3-year degree, I started to feel like I was hearing the same argument more than once in the philosophy, and the physics kind of got more interesting. We started doing quantum mechanics, we started doing collective effects. And so I gradually sort of steered myself back in the direction of physics. I ended up doing a PhD in theoretical physics, and I've basically worked in the field since. And what I'm interested in really is kind of a broad topic, and you'll see how this applies to the synthetic biology question as well, which is the whole question of collective behavior. So I take let's think about a system which is composed of a large number of simple, maybe identical particles, and let me put them all in the same space and they interact with each other in some way. And the kind of question I'm interested in is, what kinds of collective behavior might I see these particles doing together that I wouldn't necessarily have predicted based on experiments involving 1 or 2 of them. These don't have to be very highfalutin settings. So, I mean, I don't work on traffic flow, but I have colleagues who work on traffic flow. And that's an interesting example, right? So if the roads are almost empty, then the right language to use to describe the road network is in terms of cars, right? If there are only 3 cars on the road network, you ask, where are those cars? And I just give you 3 positions and you know where everything is. As you know, when the road networks get busy, traffic jams start to form. And when the road networks are very busy, actually, you don't really want to talk about where the cars are. You want to talk about where the jams are. As in, where do the traffic jams start and end? And a much more efficient description of the evolution of the road network when it's busy is actually not pinpointing the cars, but pinpointing the heads and the tails of the traffic jams. And it turns out you can work out rules by which the sort of heads and tails of the traffic jams move along the road. And then you don't really have to think about the cars anymore at all. You can just think about where the heads and the tails of the traffic jams are. And you can write a sort of efficient model for that, which contains a lot fewer entities in it than you would have had if you described the cars individually, but captures basically the same information. The sea breaking on the shore is another example. Right? We know it's made of water molecules. In principle, you could describe the motion of the water by describing the motion of all the individual molecules that compose it, but that would be insanely wasteful and very computationally inefficient. I mean, we can't do that with current or plausible future computational resources. So how do I predict what the water is going to do? Well, I try and build a theory that doesn't rely on the constituents of the water, but just uses the fact that it's got a certain resistance to shear, it's got a certain resistance to compression. I try and write a theory that just has the basic mechanical properties of the water in it. And that gets me a theory which is called the Navier-Stokes equation, or variants of it, which I can actually use then quite well to simulate waves breaking on the shore as the water flows over shallower and shallower land. Now, my actual research is not about those cases of collective behavior, but it's about cases of collective behavior where the fact that the constituent particles obey quantum mechanics is important. So, for example, iron is magnetic. I don't know whether you knew this, but in a world without quantum mechanics, permanent magnets would be impossible. So whenever you stick a magnet to your fridge, you are actually looking at a direct manifestation of the quantum nature of the particles that compose it. Permanent magnetism cannot exist in the classical theory. It exists only because of quantum mechanics. Similarly with superconductivity and various other collective phenomena that one sees in solids. I have a kind of side hustle about collective phenomena in the interior of black holes. But you know, that's not respectable enough to make a complete career out of. And obviously the techniques that you use to think about how to go from microscopic to large scale descriptions in an efficient way are actually quite transferable, and that kind of thinking has given at least some illumination, I think, as well as quite a few red herrings in the air, no pun intended, in the area of synthetic biology, and thinking not just about how to describe what's going on with the various chemicals in the interior of cells, as you know, E. coli does its thing with the extra bit of stuff you've spliced into its DNA, but also modeling things like if I put a lot of these organisms, say some of which are genetically modified and some of which are not, into a large arena and let them compete, what if anything can I say in terms of computational modeling about, you know, how often the genetically modified organism tends to overwhelm the one that was natively there, how often the genetically modified organism dies out, and what controls that kind of competition. So it's always a search for the right effective variables in terms of which to describe the problem. And the sort of animating spirit is that if you get the collective variables for the description right, the problem often becomes easier than you thought it was.
Thanks very much for that, Chris.
Thank you, Chris. I think the first question that you've received from the floor is simply whether you'd say that research has become more interdisciplinary in the last few years, both in your field and overall.
Yeah, I think the answer is it has. There was, I mean, when I was an undergraduate, there was active antipathy between, say, physicists and chemists. And we were definitely brought up to think that chemistry was basically not worth doing. And I think the chemists were basically brought up to think that physics was not worth doing. None of us talked to the biologists, so I don't know what they were taught about us or what we were taught about them. But I'm sure it wasn't complimentary. And there has definitely been a tendency in the past, I would say 20 years maybe, for this kind of Renaissance spirit of, you know, everybody's welcome to try and think about these problems and bring a new perspective to them, as opposed to the more conservative, you know, we're the experts and what could a chemical engineer possibly have to tell us about quantum computing? So yeah, I think it has rebalanced somewhat. And of course, it's interesting to ask whether that's really driven by funding, or driven by government, or driven by actions of people within the academic community, if we're talking about academic research, or actions of companies, if we're talking about industrial research. And that's actually not a straightforward question. There are all sorts of incentives and all sorts of barriers to interdisciplinary research. But I guess there's certainly been a sense that the level of quantitative control we're getting over our descriptions and our treatments of biochemical systems feels a lot greater to me now than it did 20 years ago or so. And I think that's also helped to draw people in, because the more quantitative, the more precise the control, the more possibility there is of doing something with the system rather than just trying to explain what it's already doing. And I think that's caught a lot of people's imagination in the areas of biology and biochemistry.
And I mean, of course, we will come back to these questions in more depth, but I think to follow the schedule, would we maybe like to go on to Richard, your introduction?
Okay. Yes. If I can share my screen, I will.
The first hurdle.
Yeah, at least attempt to do that. Okay. Hopefully you're seeing a picture of Heidelberg. So a brief introduction to me, I did my undergraduate degree in St Andrews. I studied biochemistry, and then I moved to London and Imperial College, where I did my master's and my PhD. And then I ran a small research group there before I was recruited to Heidelberg. Heidelberg is actually rather similar to St Andrews, an ancient old university. It's very pretty, lots of students everywhere. It's a nice bit of Germany. You should come and visit. But what I really want to tell you about is the work that I do, and it's really been my research goal over the years. This is the snappy title. We're making DNA nanovectors for gene and cell therapy.
Sorry to interrupt, Richard. It's just, are you sharing your screen at the moment?
I should be sharing my screen.
Yeah, so all we can see is a black screen at the moment.
Okay, it's the wrong screen. I'm sorry.
No worries.
Okay.
Now we can see yourself.
Yeah, you can see me. Right, I will share the main screen, and then I guess this is going to go.
Right.
Okay. Let me know if, well, you will recognize it. Is the slide come up now?
The slide has, yeah, it has.
Okay, so this is Heidelberg. That's what I was talking about. But this is the snappy title about my research. Summarizes what we're trying to do. Essentially, for me, I've been working for a long time to improve the vectors we would use to genetically modify humans or human cells. And my big ambition is to do this without damaging the cell or the person. And then it has many similarities, of course, to modifying other organisms. You don't want to damage them. You want them to produce the product, the gene product you're interested in. And so it's always been my intention ever since I was at Imperial working in cystic fibrosis gene therapy up until now to try and use better vectors, more exquisitely designed DNA vectors that we could use for gene therapy. Because most of the time people use viruses and other pathogens for gene therapy, and they have their own toxicities. And for me, if you damage the cell, it's going to be less useful as a therapeutic. And of course, you don't want to damage the patient. So gene therapy has been discussed in the literature for maybe a bit longer than synthetic biology. I think it was I can't see, there's a screen over it, 1972, I think, where Ted Friedman and his colleague Richard Roblin talked about gene therapy. And they really didn't discuss all the scientific and genetic hurdles to it, but they said it might raise some ethical problems as well as scientific ones. And that's certainly been the case. It's only just recently where humans are actually being treated with genetics for genetic disease. So it's really not that simple. You can take a diseased cell and you can add therapeutic DNA, and it's not so simple to get the healthy cell. You can also nowadays take diseased cells or other cells of a patient, reprogram them into stem cells, and then turn them back into healthy cells. And so our intention is to do this with better DNA. better vectors that don't cause damage. And that's what my group has been working on. And part of this, you may not recognize him, you might recognize his name. This is Professor Harold Zorhausen. And he was the director of my institute in Heidelberg, and he recruited me to Heidelberg from London. He got a Nobel Prize by showing that viruses can cause human cancers. So he discovered that human papillomaviruses cause cervical cancer. So the mere modification by a virus, which is, this is a particularly nasty one, but the modification of a human cell with a virus can lead to disease, and life-threatening disease at that. So it made me doubly reconsider why we might use viruses or we might use bacterial DNA as a therapeutic without considering how we might be able to improve them or optimize them. And of course, here is a cartoon showing that we can do gene delivery or gene therapy using viruses or now our non-viral vectors directly into a patient. And one question for the panel is a gene therapy, gene-corrected patient a genetically modified organism in the end? Probably. More recently and more commonly nowadays, we take the cells out of a patient, we might convert stem cells into healthy cells, or we might even take an immune cell, retarget it genetically to target and attack a tumor cell in immunotherapy. So all of these things are possible. Genetic modification of people or their cells is now more commonplace and is very effective. And our group is working to try and make this a little bit better. So I'll just tell you, we use some clever engineering to make our vectors. When I was in London, we made these things called mini circles. If you just look at this top left picture, A, most of us are familiar with making DNA and circles of DNA in bacteria and then using that DNA for some other purpose. What we might not consider is that half or more of this DNA is exclusively bacterial and necessary for its function in bacteria, but it's actually toxic to a human or mammalian cell. So if we take bacterial DNA and introduce it into a human cell, the human cell thinks it's been infected by bacteria and it responds. There's innate immunity, there's all sorts of cellular responses which can guard against that. So one of the research goals of the group in London was to make these mini circles, which had a circle of DNA which was useful in a mammalian cell, and you could remove the bacterial cells. More recently, we now use more sophisticated things. We work with a team in Nature Technology, a company, essentially without using antibiotic resistance or typical origins of replication or other bacterial sequences. It's essentially a selection marker with interfering RNA. We can get really, we get really pure circles of DNA which have no impact on the vitality of a mammalian cell. There's no toxicity. These DNA vectors are ignored by the cell. They don't provoke innate immunity or any of the other cellular receptors. And essentially we can generate isogenic cells now. We can genetically modify them without causing any molecular damage. And we can do that making huge amounts of DNA. So this is what we think might be a viable treatment, a genetic drug for all sorts of therapies. So we've made a vector and we can use it in place of viruses, in place of pathogenic DNA made in normal bacteria, and we can use it in a range of applications. And I would just like to thank, these are my team and some of the people we work with, and I'll be very happy to answer your questions now and later on in the discussion.
Thanks very much for that, Richard.
Brilliant.
Yeah,
So
I had the
we actually
pleasure
have.
of
Oh, sorry. What were you saying? So one of the first questions actually we have for essentially from what he or she has read about gene therapy, it's effective but expensive to carry out. Do you see the price decreasing in the next few years, and how could synthetic biology contribute to this?
I think part of the expense is the making material that's suitable for treatment to GMP quality. It's also more expensive if you use something like a recombinant virus, because if you have to make a recombinant virus, as any of you who have done that, you need to make recombinant DNA, you need to modify a cell line, which has to be under GMP conditions. You have to produce the virus under GMP conditions, and then you have to purify and quantify and qualify this virus for clinical use. viruses are pretty unstable. You can't make many of them. You might be able to treat half a dozen patients from a single batch. We're hoping, for example, if we use DNA vectors like my lab has produced, DNA is much more simple to prepare and produce. We can make tens of grams of DNA from a single fermentation to GMP quality. And the DNA is lyophilizable. We can store it and we can use much less of it. So current treatments are very expensive because we're still really at the prototype stages where we need to validate the vectors and validate the treatments. And there's only a handful of genetic treatments which are clinically approved so far. And they are, yes, very expensive.
Thank you. And finally, last question, is it really ethical to use gene therapy?
Ethical, yeah. I think if it's to treat a disease that somebody has and to treat the disease in a way that there's no genetic transmission, it's not like we're doing eugenics. We're not selecting for improved performance or lack of disease. We might be transiently creating a better life for a patient using genetics. If it's a genetic disease, often the most simple treatment is to replace or repair that defective gene. And most of the vectors that are currently used there's no possibility of transmission. So this is back to the discussion about how genetically modified organisms can be released into the environment without control. And I think the vectors that are currently used for gene therapy are either transient, they are limited to the host where they're delivered. And there's very strict controls about this. And it's for life-altering therapeutic use. And I think that's ethically something that should be approved.
Thank you. I think, although we do have one, I must say, in fact, two excellent questions. We'll again try to conserve those for the panel later.
That's fine.
So maybe we'll go on to our final speaker, but certainly not least, Slovena.
Hello, everyone. So I'm Slovena, and I'm from Bulgaria. I finished my undergrad degree here in Bulgaria, and then I just finished 2 days ago my master's degree in medical genetics and genomics in University of Glasgow. So as you can suppose, I don't have much research experience, but I have iGEM experience. I participated in Team Bulgaria 2018, and I also was something like a supervisor of Team Bulgaria 2019. I also was a part of the communication committee of After iGEM. So I'm really interested in the iGEM competition, even if I didn't have so much time this year. My interests are mostly in microbiology and cancer. I hope that in future I will gain more experience in precision medicine of cancer because I'm really interested in this. So I hope you have some questions for me because I don't have so much to show you. So I hopefully can answer you something about the iGEM, probably.
Okay, yes, actually, one of the questions that was simply asked before is from an anonymous attendee, and it says, it appears to me that synthetic biology is not talked about much, even within my course at university. So why is this? Because it seems like a more ethical and logical way of tackling problems.
I think this is because it's pretty novel and not many lecturers have experience in that. So maybe that's the reason. Also, I can tell you about Bulgaria because here nobody knows anything about that. We're studying only old things. So yeah, I don't know what is about other countries, but this is the case here.
Okay. Because certainly for something along the lines of gene sequencing, it maybe took the SQA 20 years to actually introduce that after it became a mainstream approach. So maybe one of the possibilities is a delay between the amount of time it takes for a new research proposal to actually matriculate into the education system. And another question we've got is, what was your actual iGEM project that you took part in?
Our project was a novel test for cystic fibrosis. The idea was to use CRISPR-Cas9 system and to make a test which is simpler and cheaper for the people, because here in Bulgaria, cystic fibrosis is the most common genetic disease, but the genetic tests are very expensive and very slow. It took something like at least 3 weeks for the results. So we wanted to do something similar as the diabetic tests, where you can test yourself. Just buy a kit and test yourself. Took a blood or saliva, and the test gives you the result in a couple of minutes. That was the project that year. And we were only 3 girls, which was really hard for us to do everything, but we somehow managed to do it, and we won bronze medal. So.
And so how did participating in iGEM influence your career plans? And do you intend to stay involved with iGEM?
Actually, first I was not sure about participating in iGEM, but I really liked the experience. It changed my point of view completely. I didn't want to talk with people so much about the science, but this was one of the major parts of iGEM, to promote synthetic biology to the people. And I really like that. I really like to see the interest in the kids' eyes when they look at some fluorescently labeled bacteria, for example, or looking through the microscope. I know it's really nice, and that's why I choose to become STEM ambassador here in UK. The idea is the same. STEM ambassadors go to schools or to some conferences and present their area of STEM. In my case, it's genetics, but we actually didn't do much this year because of the coronavirus. But yeah, I hope we would have more events in the next year. Also, the iGEM experience made me to choose my master's degree, because I was scared to go in other countries. But when I saw other teams, what they are doing, and when I compare it to the things that we are doing in Bulgaria, believe me, there was a huge difference. So I wanted to go somewhere else and to gain more experience. So yeah, it really changed my mind for everything.
Thank you. And perhaps therefore on a related theme, when you are communicating science to an audience without as much experience, what approach do you find is the most useful to convey your enthusiasm for the area?
Maybe when you show something that they can touch, that is the most engaging, I believe. And show them that this is not so hard. I can give you an example. In one of the conferences, we chose one of the girls. She was a kid, I think she was 10 years old, and we invited her and her little brother to our lab. And she could draw something with colorful bacteria, and it was so nice for her to do that, or to try to prepare something. Yeah, that's it. To give the people a chance to touch the science. This is the most engaging for them.
Well, again, thanks a lot to all our speakers and absolutely fantastic introductions, providing I think the ammunition to the next half of our panel. But I think I was going to hand over to Bradley for the next proceedings.
Yeah, thanks very much for that, Kais. And yeah, just to reiterate what Kai said, thanks so much to all of the panelists there giving your introductions. It's quite clear that we have a diverse range of expertise on board with us for our discussion today. But yeah, just before we proceed into that, I'd like to now present a short break for everyone. Before we go into the short, it'll be around 5 minutes long. But I would like to emphasise that in the next portion of the panel discussion, there will be prize giveaways at the end for the best question as decided by the panel experts themselves and also decided by the audience by upvoting a question. So again, I encourage engagement from everyone and to keep asking questions. We're now also going to launch a poll during this short break, and we'd really appreciate it if you would take the time to answer this poll. We're going to repeat this at the end of the panel to see how the discussion has influenced your opinions and perhaps if you've gained anything useful from the discussions. Okay, so now just for a short break for everyone to get a well-needed glass of water and stretch their legs. Okay, thanks very much. Oh, hello everyone, and welcome back. I hope that time gave you a bit of time to get settled again, get comfortable, ready for the main star of our show, which is the panel discussion. Again, I'd just like to encourage participation throughout this, and I think just to really begin the whole discussion between the whole panel of experts that we have on board today, we'll start with quite a general question that was asked by Sarah, and I'm addressing this to the whole panel, and hopefully someone has a viewpoint that they can address first. What, in your opinion, is the biggest misconception about GMOs to the general public? I think I'll start with, I'll aim this at yourself, Les, first, if you would like to address your viewpoint.
It's a tough one. When I was involved in the trials of the GM crops, we did an awful lot of public meetings, about 300 in total. And one of the big problems was that a lot of people didn't understand the basis of genetics anyway. And so often the discussions were taking place at a level where people really hadn't got the background to engage. And so getting the level right to discuss with people is by no means an easy option. And also it does depend on the audience that you've got. But I don't think there's a single lack of understanding that's easy to deal with, if that makes any sense.
Yeah, that's interesting. Do any others have any viewpoint in that?
I think for me, there's a kind of misconception that, well, first of all, that there are plant GMOs currently being produced in fields, swathes across the country, which I've had from, for instance, a taxi driver before that said, "Oh, those rapeseed fields, all those yellow flowers." That I don't know why, but that's what his assumption was, that because he'd not really seen fields of yellow flowers being farmed before this kind of debate started, he linked the two, even though that isn't the case. Whereas not that many people are aware that actually, for instance, vegetarian cheese is enabled by the use of GMOs. So GMOs are not used in the manufacture of vegetarian cheese, but the only way that we can get the enzyme to make cheese in a vegetarian manner is to produce it using a GMO, which is then, it's taken from the GMO and used in the process.
So maybe an extension to that question for the panel is, do you feel as part of that misconception in both genetic engineering and synthetic biology is the process of taking a gene from one organism or microorganism and putting it to another still has some of the properties of that previous organism? So fear surrounding, for example, inserting a pig gene into a plant, for example.
I've not come across that, but one of the points of view which is a more interesting one, which I've heard several times, is that by doing that, you're diluting the genetic purity of the organism. That there is a sort of a natural state, and by bringing other genes in, you're destroying that natural state. So it's quite hard to get across, and it's different to the point of view that I came across when we first were looking at GM crops, because there a lot of the fears were not about GM at all. They were about the mechanization, commercialization, and commoditization of food and agriculture. And it's just that GM happened to be coming up at a time when that gave people a chance to express those concerns. But naturalness is a very to an ecologist is a very strange thing because everything's natural. But this is a very strongly felt point of view by many people.
If I could maybe add a comment there, and I think this point about natural versus unnatural is very important in understanding people's perceptions. It's something that one often sees, I think, in looking at public response to new science, which is to assume that the new is totally different from the old. Whereas in fact, you know, we made all the carrots orange before we had any means of splicing DNA inside the cell. They did it purely by mutation plus artificial selection. And that method of altering the genetic character of agricultural stock has been around for centuries. I mean, Darwin sort of uses the term natural selection by analogy with artificial selection, which is a well-known farming technique which he expects his audience to know. So I think there's also this feeling that because it's totally new, there must be totally new risks associated with it. And so one way to suggest that maybe the risks are not totally unknown is to point out that many things that are done under this banner have been effectively done by other means before.
Well, so in extension to that, and by the panel, how hard do you feel it's been to actually convince politicians that GMOs are a force for good or can be used for good? So maybe start with Louise.
I think the UK, in comparison to other countries in Europe, is actually very supportive of the use of GMOs. I don't think it's been argued for a number of years now that perhaps synthetic biology was the wrong term to use because it introduces the word synthetic and that's going to turn people off. I think politicians, because they're wanting to see an economic benefit, if you can explain to them the economic benefit or the more sustainable approach, then they're open to discussion. But politicians are elected. And therefore, if you don't take the public with you on this scientific journey, then it doesn't matter what, convincing the politicians isn't going to do anything because they can make their own personal judgment, but they want to be elected. That's their job. That's what they want to do. So they want they want everything that the policy that they support, they want it to be supported by the people that elect them. So you can't kind of, it's not one or the other.
Yeah, I think there is a greater demand perhaps in Britain for, supermarkets are stocked differently there. British people seem to demand strawberries on the shelves every month of the year. And that's not agriculturally possible in Britain. Germans don't do that. Germans will have strawberries in the strawberry season, they'll have the asparagus in May, and it's finished. And there's not the requirement to import foods and generate food like this. So I think Brits, although I think scientists and the people who did this in the early days did a poor job of educating the public. I mean, I had discussions about these things and, you know, even a family member says, I don't want to eat DNA. Because they didn't really understand that there was DNA in tomatoes anyway, but now we were putting DNA into tomatoes and that was somehow foreign. So I think the whole discussion about genetics and genes and now viruses and the rest is people are much more educated and aware of these things. And I certainly think politicians will be swayed by consumer demand for cheaper, better, riper, all year round fruits. And I think that's sadly how they're influenced, and it's not necessarily for the right reasons, but that's public opinion is their main driver.
So alongside the same question, And then also in terms of how this can be perceived, at least in politics. What areas outside of agriculture, and for that matter, food and recycling and health, so the areas that you've described, could benefit from synthetic biology in your opinion? And for example, the one that Michael gives is, are self-repairing materials possible in the foreseeable future? Should we maybe start with Slavena this time?
Yeah, I'm not sure about this question really, because I don't have so much knowledge in GMO. I've used it only in my undergrad for a little time, so I can tell you only in the medical point of view, but not the other things.
And so in medicine, which areas have you seen expand most in the last decade, at least using gene editing and synthetic biology?
It can be used for many diseases, and especially for precision medicine for different diseases. Actually, gene editing is a really good way to do that, because they can point the exact patient in the exact condition. So I think this is.
Yeah, I think certainly in these current days, everything we will hear about treatments, vaccines for coronavirus and for treating COVID-19 will be genetic engineering. There'll be synthetic RNA molecules, synthetic DNA molecules used as vaccines, recombinant viruses will be generated to express coronavirus epitopes for vaccinations. And the use and the utility of recombinant genetics and genetic engineering will be at the fore, certainly when it comes to the treatments and vaccines. So that's one medical application that's not gene therapy or cell therapy, but it's going to be very pertinent and hopefully in the coming months.
Maybe if I can add some rather non-expert thoughts there, I have considered over the years of iGEM what range of projects we tend to see, and I think there are two main categories really, and then some exceptions. One category is using genetically modified organisms to achieve things inside or in relation to other biological things. And that sort of makes sense, given that they're the right kind of squishiness and they're the right kind of size to kind of float around and interact with other biological organisms. And then there are things like the stuff that Louise was talking about of secreting things that are smaller than themselves. So cells are actually quite good at picking things up and carrying them around, and they can be talked into picking up things that they don't normally pick up and then spitting them out later. And that kind of application is sort of clear for things that are smaller than them, but not very clear for things that are bigger than them. So I see the two sort of applications really as being kind of biological and nanoscopic in some sense. And obviously lots of nanoparticle processing leads to large results. So that they can be very useful that way. In terms of self-repairing materials, I mean, I think I wouldn't say that would never be possible, but one of the difficulties is that biological matter does tend to be kind of soft and squishy, and often what you want for these more physical applications is something really quite tough. So if you just get biological material to fill a crack, it won't be structurally up to the job in most cases of actually holding anything together or being as resistive as you want it to be, which is not to say those applications don't exist, but they're less naturally adapted to what these synthetic biology chassis are made of, and I think they'll need more development before a real kind of a really kind of world-changing application in that direction comes about. But happy to be contradicted if other people have patents up their sleeves.
And at the agriculture and environment end, the big goal at the moment is to increase the efficiency of photosynthesis. because at the moment it's only working around about 3% efficient. We know we need to produce more food, basically with the same resources, in an increasingly more difficult environment to grow things. And so if you can make photosynthesis more efficient, and ideally couple that with making the plants and crops more tolerant to drought, more tolerant to salt, poor quality soils, etc., these are big gains. And the early field trials on photosynthesis show that you can make quite a difference just by tweaking how the plants manage water conditions and gas exchange. And so this is not a pipe dream, it's going quite well.
So to that end, and maybe actually, Heng, would you like to answer this question yourself? I think I can enable you to talk. So I think you have an excellent question. So you should now be able to talk if you'd like. If not, I can ask.
Yeah, can I be okay? At the start of the talk, it was mentioned that synthetic biology arose from the idea of trying to modify the behavior of organisms, of searching up a particular gene and entering them. And Mr. Harbottle has been talking about how developing methods of making it more low cost, for example, in the area of medical treatments. How would the panel envision that once these technologies become more low cost, how would these be controlled? How would we be able to control how people use these technologies?
Would we maybe like to start with Louise?
Okay. Yes, my thoughts are still with the last question, which isn't very helpful. Okay, so I mean, this isn't really how I see things, which isn't helpful with how I'm going to be able to answer this. So I do appreciate that, yes, synthetic biology, one of the goals is to be able to freely modify the behavior of organisms. If development were to be successful and become more low cost, I mean, the thing is, some synthetic biology advances are purely to make things more low cost, to open up and democratize the lab science that people can do, so that it's actually the enabling tool, it's an underpinning tool, so that there's so many different applications that people can, by being different and inclusive, everybody can explore them. How do we envisage the control of how synthetic biology is used? I'm not. Do we mean how we're controlling the cells by Wi-Fi, or do we mean how we should control the use of synthetic biology, and it shouldn't be democratized, and it should? I'm not quite clear on what the question is to start with.
Can I come in? First of all, I think this is an excellent question, and from the point of, so I want to make it clear, I'm speaking personally, not on behalf of EFSA. The current regulations assume that basically the development processes are expensive. And so there's not that many proposals come through at any one time. And the amount of evidence that comes behind each one is considerable. So to put an application to release a GMO in Europe costs typically around about £300,000. It's not at all clear how we'd be able to cope with regulating lots and lots of very cheap applications, often by organizations and people who simply do not know of the evidence requirements to get them through for environmental release. What's more concerning is the very real possibility that although everyone in the public space is very good and we're all in favor of the future of humanity, etc., etc., there are genuine concerns that some of this work is being weaponized. Not necessarily within the, it might be by terrorists, it might be by governments, and the cheaper and easier this gets, the more likelihood that becomes. So the issue of control is a very difficult one and a very real one, and it's an excellent question.
Thanks very much for that, Les. You know, I have a question myself on how to follow up on that. You mentioned that the easier it becomes, the more widely applicable they become, then the more dangerous they become. And I think that's true in nearly all forms of technology. Especially, I use the example of computing power with the introduction of, you know, quantum computers that are now becoming more and more powerful, and that's posing threats to traditional encryption services. And to deal with that, we're now trying to use quantum encryption. And, you know, so how do you think we can deal with these threats that might be posed by more wide application of GMOs? You know, can we have a GMO police service?
Anyone that's come up with a good answer to that one.
Okay.
I do think that we have to remember that society has been dramatically changed by developments in areas such as artificial intelligence, Facebook, in the politics of the world has dramatically changed because of certain scientific developments. Biosecurity has been flagged as a concern with regards to synthetic biology for an awful long time. Garage synthetic biologists that can go have a play, hobbyists. If you actually talk to the FBI that are probably going to be there represented at iGEM, they will tell you that this is not the bit they're concerned about, actually. And it's not, it's just, I think it's because the community of synthetic biologists has tried to ask these questions. It's tried to both adopt the precautionary principle and to this foresight, this responsible innovation, the responsible research, so that it looks at this and it opens it up and it has this conversation, it has this dialogue. It's not just about educating people, it's about talking to them. We want to work with all the different areas of the publics and co-determine what kind of world we want to live in. What we don't want is some big name synthetic biologist, like basically coming forward, giving us Facebook and then going, now I'm going to change the whole future of synthetic biology and the world.
Okay. So with that, Louise, and this is Heather's question, just following that up, is do you know of any times in the past then that GMOs have actually affected the environment? Is there any examples that you could give that causes this fear of what they could do if they become more widely applicable?
So with regards to bioremediation, when you're actually using a naturally occurring bacterium, when you take it out of its environment and in the lab it does the job that you want it to do. If you and you've been working on it and you've studied it and everything, and then you want to use it in a new site. This is a naturally occurring organism that is well studied. It still doesn't work. Bioremediation, one of the real challenges is actually being able to take a microbe from one environment and it's still work in another, because it doesn't, because that's not the conditions that were optimum for its growth, and it has to compete with everything. When you actually, the engineered organisms that we're talking about. Certainly like Lab E. coli, it's been so messed around with. It's the wimpiest, so it works lovely in the lab when you throw a load of antibiotics in and make sure everything else is dead. But it is the wimpiest thing. It can't outcompete anything. So there have been no incidences that, and I have tried to find any, I am not aware of any incidents where there has been an accidental release of a GMO that has successfully managed to establish itself in any way. It doesn't happen. With regards to actual GMOs that are being used in field trials, plants that are being examined for and being cultivated, I'm not the person to talk about that. I don't believe that there's been any reports of them being problematic in any way. But that's definitely not my expertise, and that should be Leslie.
Yeah, if I can come in there, there's been no records of harm that I would recognise as harm as an ecologist. And that says a great deal about the power and the effectiveness of the screening techniques that we've got. And also that a lot of the screening takes place before it ever gets to the regulators. But there has been commercial harm. For instance, where people have looked hard and they find traces of GM pollen on an organic crop or inside organic honey. It doesn't make any difference to its quality as far as I'm concerned, but to a lot of the customers, it does. It is no longer pure. And so there are issues like that, but in terms of has there been any release of GM that's made a real difference that could be measured in terms of plants, animals, or pollution? No.
Yeah, so sorry, just elaborate on that, Les, and sorry to interrupt you, but what does pure mean to you, and what does it mean to the public, and is it valid what they see as, you know, organic versus non-organic? And
it's very difficult one to get your idea across, but to give a flavour of how extreme it can be, I had one meeting in Scotland where I had this vegan was very concerned that there would be GM pollen on his strawberries and that the pollen might include genes from an animal. The idea that pollen is floating around all the time, the idea that many genes from many species are shared across everything had not occurred to him. For him, he has a view of the purity of his product, which is simply not feasible and not delivered at all. But nonetheless, the view is sincerely held and the GM challenges that view.
yes, well, how can we, I know, and this is coming on to the main topic of this forum, is how can we educate the public more on this sort of thing? And
with this guy, I gave up and offered to buy him a drink. I couldn't think of anything else.
But I think you've got to be careful about saying it's about educating the public. It's actually, as a scientist, it's being open, and this is what iGEM is all about. It's about having these conversations through the whole process of research and innovation, and actually changing the way you do things so that it really will have something that comes out the other end that is of benefit, because people want it. People want it to benefit them. They want to know the result. They want to invest in that process. It's not about you saying, this is going to be good and it's going to work because of it. So it's a process, it's a journey, and iGEM is the reason why it's such a fantastic tool for learning about not just synthetic biology, but actually good research practice, is because it takes you through this.
And if I might just add to that, I know it is an important part of getting beyond the feeling that we just have to tell the public what's right, is to be appropriately self-critical. If a member of the public says, I heard a scientist on the radio telling me that this was safe, and I remember the thalidomide scandal, and I remember that these people made the bomb, and I remember when they were painting radium on watch handles to make them glow, and that was all fine. And one has to say that it is not unreasonable, I think, of the public to say, if the game is believe what the scientists tell you, I can think of some cases where that wasn't a good idea, and how do I know that this isn't one of them? So I think education really has to be about what the word literally means, which is drawing out. It has to be trying to develop people's cognitive capacities and their awareness of what we actually do, to the point where they can make their own judgments about whether they are confident in the process, and therefore whether they are confident in the results it produces. And hopefully in the process of that, they would conclude that we are a bit better about process than we were when we were painting radium on watch hands to make them glow. But that feels like the important thing to get across. The more that science seems monolithic, the more we're actually identifying ourselves with people who in the past have done quite a lot of harm. And it's important to be more nuanced and to make it a discussion of how things used to work, how they currently work. Talk about the process, I think.
Thanks for that, guys. Okay, we have a question from Sarah, and I'll just go into allow her to talk so she can ask herself.
Yeah, which of my questions are you referring to?
Your question on the regulations and the use of GMOs.
Yes, so countries who have strict regulations on the use of GMOs seem to not have a negative stance on RNA vaccines. At least I haven't heard anything about any countries being particularly against the RNA vaccine developed against coronavirus. So what is your opinion on how the use of RNA, for example, in the form of vaccines, is different or similar to GMOs?
I'm going to ask yourself, Richard, this as an expert in the...
Oh, as an expert. It depends whether we define as a patient or a person being treated with an RNA. Are they really going to be a genetically modified organism at the end? Probably. I mean, most of our drugs, or many of our drugs, are based on recombinant enzymes and bacterially produced products. And there's certainly some of them. The gene therapy, cell therapy is, a different angle. I think synthetic RNA injected into a person that can exist only transiently and produce an epitope that might provoke the immune system to fight a virus. I think in the end, that's not you may have a transient muscle cell, which is genetically modified, but for days or a week, if you're lucky. You will have a different immune profile afterwards. But I don't think that muscle cell or the patient whose arm it was injected into really is a genetically modified organism then. Some permanent modification with genes or viruses that are integrating into the genome and this person is permanently changed, that might be a different discussion. Transient RNA and DNA vaccines, I'm not really sure that it certainly doesn't cause any persistence, and it wouldn't be passed on to future generations. It's not going to change the person other than their immune responses against the epitope encoded by the RNA. But how was that RNA produced? It was obviously either synthetically produced or produced in a microorganism. I'm not really sure. I think for vaccines and transient treatment, it's not much different from using a recombinant enzyme, I think.
Thank you.
Perhaps that goes on to another persistent question in this field, and that's really maybe a difference between a germline change to the genome versus one that is done in a somatic cell. And perhaps the question is, what are the views of the panel on the use of germline genetic engineering, both in terms of crops to GM crops, but also in terms of future remediation, future therapy? Perhaps
who's your expert?
Leslie, would you like to take this one?
Well, in crops, it has to be in the germline, because the whole point is that you make fairly small batches of the new variety, which have to be bulked up. So if it's not in the germline, that wouldn't work. So you have to make enough seeds for people to plant it.
In which case, would you suggest that essentially a terminating mechanism or a kill switch is necessary to include at a certain generation?
There's been a lot of concern about Terminator genes, with people arguing vehemently that they're great and arguing vehemently that they're not required. In practice in commercial agriculture, they don't arise, because what people tend to plant are what are called F1 hybrids. So these have the hybrid vigor for two different lines, and you can save the seeds for these if you like and grow them, but you do not get the homogeneity of the crops. Vigor goes down, diversity goes up. So why would you even try? So even for small scale agriculture, the idea of saved seeds, etc., etc., is a very inefficient way of doing it compared with how seeds are produced at the moment.
Yes, we've noticed that Michael, you're a very prolific question asker. So perhaps would you be interested in actually asking your last question there to Leslie? I think you should be able to talk now if you'd like to. Okay, well, this question was essentially that at the beginning, GMOs can affect the biodiversity within an agricultural setting, primarily through which herbicides are used. However, have you seen examples where GM crops may directly lead to an increase in species diversity?
Hi there, sorry, can you hear me?
Okay.
Yeah, we can hear you now, Michael.
Okay, brilliant. Sorry, I'm just trying to remember which question it was I asked.
It's the last one we were addressing there on there.
Okay, yeah. I think the question was just regarding GMOs and the biodiversity within an agricultural setting, which, you said it was affected primarily through which herbicides are used. I just wanted to ask, have you seen any examples where GM crops may directly lead to an increase in species biodiversity?
The best theoretical example is if you use GM crops that contain pesticides, and those pesticides are targeted only at those insects that actually attack those crops, which in principle means that you need far less pesticide elsewhere in the environment. So that should be a win. With the new ideas coming through on gene-drive mosquitoes, that could be a particularly big win, because that could result in the reduction of use of broad-spectrum insecticides. But it's always more complicated than that. So one of the concerns about the gene-drive approach is if the gene-drive approach fails, you would still need something else to fall back on, which may be the insecticides anyway. So often the benefits and the problems aren't as extreme as one might feel.
Michael, I hope that answers your question, and while you're still talking to us, and you can, feel free to ask your other questions.
Oh yeah, sorry. Thank you very much for answering that, Leslie. It was good to hear. And I think the other thing that I said that you might have asked me about was about, reading a scientific journal can be quite a daunting task, especially given the level of, we talk about business jargon, but scientific jargon, especially as a layman. So I was just wondering if there's some feeling of responsibility within the scientific community to make the information more accessible in order to allow the public to be able to kind of critically think through the benefits or the harm of specific research.
I mean, maybe I could start on that one. I definitely have a sense of that responsibility, and I think that the reason it doesn't happen more is that there are kind of structural problems about making it happen. The way that the pathway, at least to academic scientific research, is set up is very specialized. And it's quite competitive as well. I mean, there are many fewer academic jobs than people who would like to have academic jobs. And because of that, there tends to be a reward for quite ruthless focus, which tends to bring with it quite a lot of specialization. And so one can end up that way without really trying to, in the position where the people who've been really successful in academic research are the ones who have deliberately specialized, carved out a niche, become the best person in the world at doing certain types of computational calculation on, you know, electrons in spin ices or whatever it is. And that set of skills doesn't necessarily mean that they don't have the ability to communicate more broadly or to get their work out to a larger population, but it certainly means that they will have had their attention distracted from developing those skills, because all the strong incentives were to stay specialized and, you know, to advance the work of their research group and to publish in journals. And this is sort of what they get recruited on. So I think there is absolutely that responsibility, but I think there's a real discussion to have about how we get from where we are to an academic system that properly rewards that kind of engagement. Yeah, there's more to say perhaps, but I just maybe I'll highlight that question and perhaps others have comments.
Yeah, I think as an academic, you're right. It's actually a daunting task to write a scientific manuscript. It's not normal language, and you have to have a really strict focus. And the editors and the reviewers will be very tough with you if you don't write appropriately. But I think we need to, as academics or researchers, we need to somehow be shielded and helped by our institutions. And then also by the bigger bosses, the politicians, who need to say that what scientists say is worth listening to and give us a forum to discuss things with the public, rather than dismissing our research and our ideas that perhaps some of our current politicians do. It's tough. We have to really publicize our work to our peers and the reviewers and the editors of journals. And in some ways, of course, it would be responsible of us to communicate directly with the public, but that in some ways is not our job. Not to directly do that without the infrastructure being in place to enable it. It's really difficult, but it's very important.
Well, I mean, I think it's very dependent on what field you're in and how perhaps interdisciplinary it is. To be a good interdisciplinary scientist, you have to be able to talk across disciplines. And in doing so, you actually lose, or almost automatically, you lose a lot of the very specific terms from the language when you're explaining something. That might be those specific terms that a non-specialist wouldn't understand. Or alternatively, you know, just through talking about it all the time, you actually know which are the problem terms, so you can pause, explain those, and then carry on with what you're doing. I'm very, very much committed to making sure that the research that we do is communicated to anybody who's interested. So we've got short animations on my website that are put together to be as easily understandable as possible. For the European Federation of Biotechnology, the division that I'm in as well, we've got a communications section, which is a group of PhD and postdoc volunteers who are actually also committed to trying to have an international journal club where they take some of the leading papers as they come out and be able to communicate them in a shortened format. So they're not even necessarily relying on the research group itself, who's put that research out there to do that translation. They want to try and identify those key papers and then help others to understand them. I think iGEM's fantastic at being able to train the next generation of synthetic biologists to talk across disciplines, to talk to both experts and industrialists and the more general publics, get involved in schools. I think it encourages everybody to take all these aspects of what I believe are different facets of an academic career to actually train you in those and find out which bits of it you enjoy.
One thing that there was a point made earlier on about it's not necessarily the scientist's job to do this. I've done quite a lot of work over the years with a group called the Science Media Centre, and that's a unit. It was started in Britain, but actually they have parallel units across the world, which is to try to get journalists involved in the science so that at least they understand what information it is that they're trying to report on. And that's been really very successful. So the quality of the science reporting in the UK in this area has picked up a lot over the last 20 years. And so the work that I was doing with them, we actually had sessions with journalists before our results came out, so that they had an idea what context to give it, what it actually meant, and so on. There's a lot of people out there involved in trying to build up the engagement of people with science. The real challenge is a lot of people out there aren't really that bothered about it. And so they're perfectly happy with their opinions as they are now, right or wrong, and yet it's still those opinions that will influence purchase patterns and political decisions.
Thanks very much for that, Les. And thanks for everyone. Just before we kind of wrap up here, there's another question from Michael, and I'm gonna open it to both Slovena and our other iGEM panel, iGEM team members on here today, like Polly and Lawrence, because it's very relevant to them. So Michael says, as someone with no experience with iGEM, how difficult is it to get involved with the competition? Do you really require solid knowledge base first, or do you learn as you work within the team? So what does our team think about that? And Slovena as well, what do you think about that?
You don't need much experience actually. In my team, the other two girls didn't have any experience because they were just second year and they didn't have some lab work until then. But with my help and the help of the supervisor, they managed to work very good. So you just have to have the interest in this.
Right. Lawrence and Polly, do you think you have any opinions on that as our team members this year?
Yeah, I think because myself, I don't have much experience. I hope to gain some experience in lab work in the wet lab, but unfortunately I couldn't. But I think that there is something which everyone can do and which everyone can assist to the team. And just you have to have a good team and establish good relations with them, and then you find your job and how you can be most useful.
Yeah, I would agree. I think as long as you've got an interest sort of in sort of synthetic biology, not even synthetic biology, but just biology in general, you just pick everything up along the way. And yeah, I think as was mentioned, you don't have to have a biology background. There's loads of different roles, like in our team alone, that yeah, sort of human practices, sort of more computational modeling side of things. So yeah, just a multidisciplinary approach for sure.
Yeah, I would even reiterate that as myself, a physicist and with no real background in biology, except from a small work placement with Dr. Richard Harbother herself in Healdsburg. So yeah, I didn't have much experience with biology before either. And it was just something that interested me and I wanted to get more involved in.
To top that, I really think iGEM is so uniquely useful because it really integrates all the areas that this panel has really discussed, both, as Lawrence mentioned, in terms of the human factors side, so how your particular research might actually affect people in the general public, how it goes, and this actually involves also talking to our end users as well, all the way down to the lab side and the lab modeling. So it really is quite diverse and caters to anyone on the team. And it really is a learn as you go process. I think from this year's St. Andrews team, we've just learned every week essentially.
Yeah. So we're coming to the end of our time, and I think we need to wrap up now. So I'd really like to thank everyone, the panelists and everything, for providing such a riveting discussion, and I really hope everyone's enjoyed it. Before we go, we mentioned that we had some prizes for the best questions throughout the panel discussion, and also a little raffle for everyone who came and attended today. So I think I'm going to hand it over to Yashwan right now and Sue to announce the winners while we give the panel some time to think about what their opinion of the best question was.
And just for those from the floor as well, just if you'd like to vote, as you can see, you'll simply see a thumbs up that you can simply click to the question that you'd like to vote with.
Right, so we have 3 giveaways today. So the first one that I will present is what we named the 3 C's. And we will put all the attendees into a raffle, and the winner will be able to choose either a hamper of chocolates, cheeses, or chili sauces. So Yashwan, she will be running the raffle.
Yeah, I will share my screen now. Yeah. So it's in the alphabetical order now. And I'm gonna randomize this. And then here we go.
Wow.
Quite the visual impact.
Oh. Congratulations.
Yeah, we have a winner now. Congratulations.
Right, congratulations, Michael. So please type which hamper you would like. I will
Or just tell us, in fact. I think you still can talk.
Yeah, I'll definitely take the cheese, please.
Good choice.
Brilliant. And Michael,
Is
yeah,
it
if you
GM cheese or not?
if you could send us your email either via the chat or get in contact with our email, whatever method you prefer, and we'll organise that for you.
Perfect. Thank you very much.
And then our second giveaway is a paperback copy of Nesta Carey's Hacking the Code of Life. And as stated by the panelists, they would like to give this book to Sarah, as she had the best question. So Sarah, please type your email to us, and we will get in contact with you. And then our third giveaway, it will be a paperback copy of a Kraken creation. And this will go to the individual who asked the best question as chosen by the audience. So if everyone could please give a thumbs up to which question you think has been the best one, and we will announce the winner shortly. we have a tie currently going on right now between Yatish and Sarah. So Yatish's question was, I know that GMOs are meant for the better of humanity and to improve the methods of us using energy, but as seen in the past, any new technology or breakthrough that we make to solve a problem have raised many other problems. So how confident are we that GMOs will not do any harm to the ecosystem? And Sarah's question was, countries who have strict regulations on the use of GMOs seem not to have a negative stance on RNA vaccines. What is your opinion on how to use the RNA? So, okay, we seem to have a winner, and that is Yatish Ariel. So if you could also please type your email and we'll get in touch with you.
Right. Well, thanks very much, Sue. And again, thanks everyone for coming. I'd just like to finish by asking one final thing of the audience, is that at the end of, once we end this webinar, there will be a small feedback survey. And if you could take 2 minutes to fill that out, it'd be of great use to us for planning future events like this and hearing your feedback. So really hope you enjoyed everything today. And I think we're out. I think it's a bye from me.
Thank you.
Sounds almost like the university challenge.
Yeah, thanks to everyone. Good questions. Thank you.
Yeah, indeed. Thanks everyone. Nice to meet the people I didn't know already as well.
Yeah, nice to see you.
Thank you.
Bye.
Enjoy the sunshine.
Okay, well, I don't have any theme music to end, but I'll be, you can
I guess we'll just go.
We'll just go.
Yeah.
See you.
Bye-bye.
Thank you.
Bye-bye.
And enjoy your hamper.
Bye.
Bye.